Bismillahirrahmanirrahim. Assalamu alaikum. My name is Kiran Aslam. My department is chemistry and my ID is 005. Today I am going to present presentation of analytical chemistry and my presentation topic is accuracy of pH measurement. So first I want to tell you presentation layout which I am gonna talk you in accuracy of pH measurement. First we will discuss what is pH, then I will tell you its importance, and in accuracy of pH measurement we will discuss some methods, limitations, precautions, and then its applications. So first moving toward what is pH. pH is a scale which we use to specify the acidity or the basicity of an aqueous solution. Acidic solutions have a higher concentration of hydrogen ions, so they will have lower pH value as compared to the basic or the alkaline solutions. pH stands for the power of hydrogen or the potential of hydrogen. The pH scale is logarithmic and inversely indicates the concentration of hydrogen ion in the solution. This is because the formula which we use to calculate pH. For example, we can see from this equation. The pH is equal to the negative of the base 10 logarithm of the molar concentration of hydrogen in the solution. So it gives the inverse value, means the acid has a high concentration of hydrogen ions, so they have less pH value, less than 7. And the alkaline solution have pH greater than 7, means that they have lower concentration of hydrogen ion. Importance of pH. pH is an important quantity that reflects the chemical conditions of solution. It can control the availability of nutrients. For example, in the soil, we can calculate that which type of nutrient is required to the plant for the healthy growth, biological functions, microbial activity, and the behavior of the chemicals. For example, our blood worked at a specific pH, and we determined that pH. Next is the methods which we use for the pH measurement. The first is indicator paper or pH paper. It is an inexpensive method that provides an indication of acidity or alkalinity but not an accurate measurement. With the help of indicator papers or pH meter, we can't determine the concentration of the hydrogen ion. We just do difference between the two solutions on the basis of their color. For example, from this figure we can see if the solution is highly acidic, it will turn to the dark red color. And if the solution is alkaline, it will turn to black color. So this is how we indicate the different solutions with the help of indicator paper. Next method is pH meter. Why we use pH meter instead of indicator paper? If you are using litmus paper, none of this matters. The basic idea is that the paper turns a slightly different color in solutions between pH 1 and 14, and by comparing your paper to a color chart, you can simply read of the acidity or the alkalinity without worrying how many hydrogen ions there are. But a pH meter somehow has to measure the concentration of the hydrogen ions. The construction of the pH meter. In pH meter, there are two electrodes. The first is reference electrode and the other is glass electrode. We will dip the reference electrode in a solution whose pH value have to be found. And here is a voltmeter or a meter from which we determine the potential difference that creates between these two electrodes. So next is the working of the pH meter. When we dip the reference electrode in the blue solution and the glass electrode in the solution of the known pH, the ions from this blue solution will transfer into this solution and will replace the metal ion. So this process is known as ion exchange, and when this process takes place, it will create a potential difference which we will read on this meter. If it will give lower value, it means that the solution is acidic and it has a high concentration of hydrogen ion. But if there is less number of hydrogen ion, It means that the solution is highly basic and it will give highest value of pH. So this is how we determine the pH with the help of pH meter. Accuracy of pH measurement is governed by the accuracy to which hydrogen ions activity of the standard buffer is known. But the accuracy is not better than 0.01 pH unit due to certain limitations. The first limitation is the activity of single ion, and the second one is residue liquid junction. Activity of single ion, we can't control and calculate the activity coefficient of single ion. Residue liquid junction. At the point of junction, as we can see from this figure, at a point of junction where the two solutions meet. Here it creates a potential difference because of the ionic transfer, and this potential is known as liquid junction potential, or we say it is diffusion potential, which is non-equilibrium potential. So the liquid junction potential limits the absolute accuracy of measurement of pH of an unknown solution to about 0.02 pH unit. So it is necessary to remove the residue liquid junction. How to remove liquid junction potential? There are basically two methods. The first is to put a salt bridge consisting of a saturated solution of potassium chloride and ammonium nitrate with a lithium acetate between the two solutions constituting the junction. So it will minimize the liquid junction potential by maintaining the ionic transfer process. And the second method is to minimize the pH and composition of the solution as near as possible because it is created due to the difference in the composition and the pH of the solution. So we will try to keep their composition and pH as near as possible. pH measurement of two similar solutions. It is also possible to discriminate between the pH of two similar solutions with the differences as small as 0.004 or even 0.002 pH unit, but this is not better than 0.002 pH unit because of the liquid junction potential. For example, if the pH value of two blood solutions are close, we can determine their difference accurately to 0.004 pH unit. If the pH measurements are made at a temperature other than that at which the standardization is made, other factors being equal, the liquid junction potential will change the temperature. For example, if we increase the temperature from 25 to 38 degrees Celsius, a change of 0.76 millivolt will be detected. Thus, for every accurate work, the cell must be standardized at the same temperature as the test solution. Here are some precautions to perform more accurate pH measurement. So the first is the pH measurement and the solution temperature. The pH value of a solution has inherent temperature characteristics. For the pH measurement, the temperature shall be controlled as accurate as possible. And if you are using a compound electrode with a built-in temperature compensation electrode, which will measure the temperature of the internal solution of the reference electrode, then keep it in such a way that it reaches the temperature of the sample. Precautions before immersing the electrode in the sample. Fill the reference part sufficiently. This is the reference part. Fill it sufficiently with the internal solution. This is the right way and this is the wrong way for the filling of the reference part. And then when bubbles are formed in the internal solution, remove them by shaking the electrode. Use a thoroughly rinsed electrode to avoid the contamination. Then, precautions after immerse the electrode in a sample. When you immerse the electrode in a sample, make sure that the surface of the potassium chloride solution shall be higher than the sample surface. So this is the potassium chloride solution and this is the sample surface. So its condition should be higher. Its surface should be higher. This is the right way and this is the wrong way. Remove the bubbles if it adhere on the electrode surface and the position of the temperature sensor should be lower than the sample surface. This is the temperature compensation electrode. Its position should be lower. Sorry, this is the right way and this is the wrong way. Here are some actions for more precise measurement. Keep the temperature constant. Perform the measurement in the sealed box in order to avoid the outside air influence. Use an airtight measuring container in order to avoid the outside air influence. Rinse the measuring container with pure water and then rinse it with the sample again. Stir the sample slowly during the measurement. Applications of pH measurement. The measurement of pH is used in producing the ink on the page, the color in a shirt. We can develop color of our desire. For example, from this figure we can see if the pH is 6, its color is purple pink. But if we want a deep pink color, we will increase its pH value. And if we want deep blue color, we will decrease its pH value at 4.5 unit. It is also used in producing the shine on a car bumper, and it is also vital to each of us in our bodies. pH measurement is also very crucial in agriculture industry for soil evaluation. Major crops require alkaline environment and hence pH measurement becomes necessary. It is also useful in food industry, especially for dairy products like cheese, curds, yogurts, etc. It becomes mandatory for the chemical and pharmaceutical industries. It has a significant factor in the production of detergents. It also helps in monitoring water treatment plants and RO water purifiers. So that's all from my side. Thank you.